In this video, I'd like to talk about some of the discoveries that have been made. For example, most of you know that Blakburn, Greider, and tstozak actually discovered telomeres. And so the shorter the telomeres are, it leads to DNA being exposed at the ends, which leads to genomic instability and cancer. And so I would like to talk about something else right now, which is a big discovery, and those are senescent cells. These are like zombie cells, which are not awake but not dead as well. And you can actually see them with beta-galactosidase, which is a stain, which stains it because senescent cells tend to have these markers. And they stain blue. And so therefore you can actually see in early age, the cells look white, pale blue in middle age, and royal blue in old age because more senescent markers. The cells are becoming more and more senescent with older age. And these senescent cells actually release inflammatory cytokines and pro-inflammatory molecules, which attract macrophages, and of course leads to heart disease, dementia, and other age-related conditions, especially cancer, because they release these pro-inflammatory molecules that promote cell growth and division. And in mice, actually, there was a study you can actually destroy these senescent cells and increase their lifespan by like 20% or so. And that gives them better heart resilience, no heart disease, less kidney disease, right? All that stuff. It can even cure or make better fibrotic lungs, making them more pliable, reduce glaucoma, so better vision, less osteoporosis or frailty in older age, and less tumors, obviously. So the cancer risk goes down when you eliminate these senescent cells. So pretty much all signs of aging, a lot of signs, you pretty much remove a lot of disease by simply removing these zombie cells. And there's actually ways to remove them. They're called scenescnet, and lytics mean lyse, or essentially killing the cell, causing it to commit apoptosis. So senolytics are used to reverse it by essentially cleaning these senescent cells via apoptosis. And Campisi actually showed in the 1950s this on senescence, that you can actually get premature aging if you put these zombie cells into the mice. And so essentially, maybe if you can start doing it in the 30s and 40s, you can actually reduce cancer by simply taking sort of a vaccine or something to cleanse these cells out. And so now we're better at kind of preventing damage at childhood disease, malaria, no predation, technology to prevent damage from hurricanes and volcanoes, less homicides, better sanitation, all allowing us to live longer. And we actually saw there's a research that was done that rapamycin actually shown to prevent inflammatory release from these senescent cells, as well as quercetin and dasatinib, which also increased the lifespan by 36%. And these trials actually began in 2018. And this is good to know because quercetin is found in things like onions, apples, caper, kale, and dasatinib is actually found in leek and maybe even other vegetables. So we can actually consume these products to better cleanse these senescent cells. It's actually pretty cool. But senolytics is definitely an area which is under research and will be probably used in the future to allowing us to extend their lifespan. And these studies have already shown 20% increase in mice and a 36% increase by simply removing these senescent cells. And so there's actually a lot. So that's first discovery on these senescent cells, these zombie cells that I wanted to talk about. And you can actually see that by staining blue, that they increase in old age, and we can actually get rid of them to live potentially a lot longer. The second discovery is junk DNA. Is actually pretty much half of our DNA, and we have these retrotransposons, which are pretty much just viral DNA, which jump. They're like keep inserting back into our genome, and they're actually silenced by sirtuins. But if you have low sirtuins, you can't silence these kind of retrotransposon, and you get this epigenetic noise due to these DNA constant reinsertions. And it has been shown that SIR6 mutants, that if you inject the anti-retrotransposons to prevent that from happening, you actually double the lifespan, increase it twice, or by 100%. They live twice longer, but simply reinjecting these retrotransposons. And so we can theoretically, therefore, use antiretrovirals or NAD boosters to keep these retrotransposons in check. because antiretrovirals prevent the DNA reinsertions, and NAD boosters, as we showed before, are fuels for sirtuins, which keep them in check, which silence these viral DNA, these viral genes or retrotransposons, because I know really genes are junk, from jumping back into our genome, constantly reinserting. And this stuff, so that's actually pretty cool, especially for the senescent cells. If we can create a vaccine which kind of eliminates these senescent cells, or if we use these retrotransposons, if we kind of can eliminate them, we can have treatment every decade, right? For example, as a vaccine, even to keep the retrotransposons in check by injecting kind of viral, anti-retrotransposons. We can do it like every decade or so, and that's actually great. Same thing for the senescent cells. We can potentially, we can potentially have a pretty much a vaccine which kind of eliminates them every decade or so, which is pretty cool, because we can pretty much target them, right, with senolytics, based on the markers that they possess, these senescent cells. And so in 2018, scientists at Stanford actually lowered the rates in mice suffering from cancer by actually what they do is these stem cells are inactivated by radiation. So if you do like these booster shots, or essentially you prime the stem cells, you prime the immune system to attack the cancer cells. And so there's the PD-1 and PD-L1. You can actually inhibit them, making them susceptible for T cells. So a way to eliminate cancer, and this is immunopharmacology, is because cancer cells essentially don't want to get eliminated by the immune system. You can actually inhibit some of the markers on the cancer cells to make them kind of visible to the T cells, so that you can get rid of them. And potentially we can do the same thing for the senescent cells. And that's where I said that you can come up with a vaccine and kind of do it every decade. The same therapy, immunopharmacology, that was used for cancer, we can do it also for senescent cells. And scientists at Stanford in 2018 showed that, in fact, that this immunopharmacology does indeed help in mice suffering from cancer. In 1958 and 1996, there were more experiments, specifically replacing DNA from an egg with an adult frog gives you tadpoles. And in 1996, it was shown that replacing sheep chromosomes gives you Dolly the sheep. And so these experiments are very amazing, because they show that our cells still have information to be young again. And so therefore we still have this this polish, which is like the toothpaste, which allows us to kind of clean all the scratches that are present in our genome, or specifically in the analog epigenetics that we talked about previously. And so that actually comes to 1948, then where Shannon actually kind of in 1948 looked at talked about how the loss of information gives us higher entropy, which was the code kind of the 1920s, the noisy channel coding theorem, right? And she showed that you can based on that, we can potentially reset aging without the need for cloning. And what happens if the data exceeds the channel capacity or it's noisy? That's how internet works, right? With the TCP, it's relevant in information theory of aging, actually. Because if data exceeds a certain channel capacity, or if it gets noisy, we eventually have to back it up. So we have a source, which is how our egg and a sperm from our parents, which give us birth to us, gives us life. Then we have a transmitter, which is like the epigenome, which is the analog information involving the biological radio signals. And then we have the receiver, which is the future body based on those epigenetic changes that we accumulate over time as we age. But we can also have an observer, which records the original data, corrects it to restore it. And so the more cell divisions we have, the more overactive survival pathway we have. And so it gets more noisy, and so the receiver loses the original information. But if the observer can kind of record it and kind of restore it, we may be able to get that backup copy, and that's really cool. So that illustrates that without cloning, so cloning shows us it's possible, but we don't need cloning to be able to reverse aging. And then the last thing I want to talk about, is what is this corrective device? And so in 2006, you probably know Yamanaka, and he discovered 4 genes: OCF4, KLF4, SOX2, and CMYC. And these actually induce pluripotent stem cells to become any cell type. And he got a Nobel Prize with John Gurdon in this in 2012. And so these Yamanaka factors are actually transcription factors, and they can become any cell type. In 2016, Serrano and Belmont actually showed that these factors, given twice a week to premature mice, can actually boost their lifespans by 40% and give them better wound healing. So 40% longer lifespan simply by injecting these for twice a week to mice who were premature. And in 2016 as well, Liu showed that tumors actually accumulate if you give them the CMYC. And so he showed that you can restore vision in mice, literally regrow, they can actually regrow their optic nerves and then restore vision with OSK reprogramming. So we only give them 3 of the 4 genes. We give them the OCF4, the KLF4, and the SOX2, but we leave the CMYC out. And so that allows the mice to actually restore vision with these 3 Yamanaka factors, which are safe to use in mice. And so TCP/IP correction, like the essentially that corrective factors based on Shannon 1948 model of the code of the noisy channel theorem, is actually could be these Yamanaka factors, which actually can make us be young again. That's actually really, really cool. And what actually does that, we don't know. The TET proteins, which activate other genes, their transcription factors themselves, appear to have a role, but we don't know exactly how is that caused, or the whole complex pathway. And then another cool thing is designer babies in 2018 was discovered of gene editing. So in the future, parents may actually be able to give their kids the traits that they want to live a very, very long and potentially indefinite lifespans. So we talked about a lot of things. We talked about, first of all, we talked about senescent cells, which I think are really cool. And we talked about the information theory of aging, and specifically Shannon's code, and how we potentially can have a backup copy by simply making us be young again using these Yamanaka factors, which are also a very recent discoveries, as well as designer babies, which are also really, really fascinating. So I hope you enjoyed these discoveries, and I actually really enjoyed making this video. Okay, I will see you in the next video. All the best.